Well, thank you, Jason. This is such a great opportunity to share this perspective on sort of, what I call a deep dive. Deep dive to me was always something that you maybe wanted to look at, but you needed to really dive deep into the history of something to really, kind of uncover uh, uh, how things transpired. And, it's such a remarkable journey what's happened in the last, say, 200 years to the understanding of the chemistry of what's going on in cannabis. And so um, I should say that, what this class is not, because it's easy to get a shallow dive across the entire cannabis plant, but we're going to do a deep dive into the compounds and structures of the cannabinoids themselves. So, unfortunately, that doesn't allow for time to say, look at the terpenes, which could be another class, Jason. We can completely spend a class just looking at terpenes.
Oh yeah.
And why limit it to cannabis? I mean, terpenes are in all plants, so you want to look at everything. I'm not going to look at cannabinoid biosynthesis, and that's a really important area because this is how compounds are made. Again, so much work been done there. Recent reviews are available. It's beyond the scope of this. I'm not going to talk about medical applications or early use by humans because, again, just such a wide variety of topics to cover there. I'm also not going to cover production, agronomy, or extraction or anything like that that's sort of going on now. I'm just going to really look at the descriptive chemistry itself of the cannabis plant. And okay, so let's dive in here. Let's see. Okay, so these were, when Jason and I had corresponded by email, this was sort of like how Jason had laid out what we thought we would cover, which was a historical view of cannabinoid chemistry. Again, the chemistry of cannabis plant specific to cannabinoids themselves, and not ignoring the terpenes, but just focusing on the cannabinoids and really what the change that happened after some time in the 1960s, after an Israeli chemist named Raphael Mechoulam um, isolated the structure correctly for the first time and showed the world what the active ingredient in the cannabis plant was. A lot of attention was paid to that discovery, and there was a lot of insinuating, or I'm sorry, a lot of other work that followed that initial discovery that really we're going to focus on today because that's very, very important. Um, Also, what's happened recently in the modern advances in cannabis chemistry, more recently, there's been a lot of discussion about something called Delta 8. So we'll talk a little bit about Delta 8 and what that means. And also consider, what the next scientific frontiers are in cannabinoid chemistry. And also get Raphael Mechoulam's perspective on that. They published a journal article actually a number of years ago, so 21 years ago, but still that frontier is still ripe for discovery and scientific innovation. So let me start just by telling you just a little bit about myself. I'm Mark Scialdone. I'm a PhD organic chemist who received a PhD doing synthetic chemistry, working with enzymes and asymmetric induction, so making chiral molecules from achiral starting materials. From that, I spent 20 years about in DuPont Central Research, and then left DuPont in 2012 and has been consulting in the cannabis and essential oil businesses ever since. So my specialty is natural products chemistry, organic chemistry, and I'm the inventor of hexahydrocannabinolic acid, which is a new cannabinoid that you get when you hydrogenate tetrahydrocannabinolic acid, which is a naturally occurring form of THCA. So received a little bit of notoriety for that, and we're still in R&D phase in developing that for um, use. Okay, so I always like to start here because this is something that I think is a unique human manifestation of the world we live in, which is that humans create art and we create science. And I think that there's a parallel between, or several parallels that exist between the two, that involves knowing the classics. So knowing Beethoven and Bach if you're in music, or knowing Michelangelo, or knowing Leonardo da Vinci. I mean, these are the masters. These are the people who basically put form to function and really created a whole renaissance of innovation and invention. And I think it's important to understand that, because rarely, whether it's in music or art or science, do you do something new without really having a firm grasp of what's been done before you. You have to know everything that's been done before you, because that sets where the current frontier is. And so when I think about cannabinoid chemistry, knowing the classics is really knowing all of those brilliant minds that worked 100 years ago on trying to understand what is in this magical plant. And there were a lot of bright people who looked at that. And we're going to look at this today and hopefully give you an appreciation for when Raphael Mechoulam entered this field, whose shoulders he was standing on, because certainly there were people who came before them. And Raphael, I think, very humbly cites all of the people who came before them, including Roger Adams and several others that we'll talk about. But it's really knowing those classics, understanding what they mean in terms of how this has enabled our current understanding, and then using that understanding and putting it in your own brain and your own thinking process, and developing a form of self-expression, of creativity, where what you see uniquely when you hear a piece of music, or when you see a piece of art, or when you read a piece of science, how that impacts you and your thinking. And so what this class is really all about, it's going to be looking at that cannabinoid literature and knowing those classics, and understanding what that didn't always exist. This work was created, and now we have this body of scientific literature. And it's the scientific literature, what I call continuum, that is indeed the crystal ball for scientists, because we can read this literature and we could understand their thought process. We could read about their failures and their successes. And from that, we can understand what can be in the future, and how we can use the understanding of where the current frontier is, which is always changing. Every paper, every article that comes out that mentions a new scientific discovery changes that frontier and moves that frontier a little bit forward every time. And so when you look at the publication of cannabinoid related articles in the peer-reviewed literature, here's what happened after Raphael Mechoulam's discovery of the THC molecule. You could see that there's been an exponential increase to the day of work that's being done and published in the scientific literature that gives us that basis to understand where the current frontier is and where us cannabinoid explorers who are dreaming about new compounds and new types of discoveries can be manifested. So I'm going to start today with this paper. This is a paper from Sir William Brooke O'Shaughnessy, who was a doctor in the UK, who studied the medical efficacy, if you will, of Indian hemp, or gunja, as he refers to here, from Cannabis indica, the effects of animal systems and health and the utility of treatment of tetanus and other convulsive diseases, including epilepsy. So in this paper is just fascinating because he talks, in fact, here you could actually see it was like 3 kids had 10 grains. I think a grain is about, I think it's a little over a gram. I'm not actually sure. I think it's 1.047 grams or something of alcoholic extract of gunja. So, I mean, this is how this kind of scientific literature was written back then. In one, there was no effect. In the second, there was a much heavier heaviness, inability to move. In a third, marked alteration of countenance was conspicuous but had no further effect. So he does tests in animals as well as humans. And here's actually, I think, his drawing of the plant. It's just fascinating. This paper, and I think it fascinated a lot of the established Western medical community, who again was opening up trade routes to the Far East, and certainly looking at other medicinal plants as well as cannabis. But to read this paper is really just fascinating, because I think what you take away from it is that not only could it help with cholera, I guess cholera was rampant in London at the time, but also the distress associated with other diseases, including epilepsy. So just very, very fascinating that that this paper started, I think, a lot of interest. And then after this paper, which was published in 1843, there was a lot of work by others, including Schlesinger, Decorative, and others, who were looking at the extracts and trying to figure out what exactly was in the cannabis plant. In the 1880s, there was a group that considered that it was an alkaloid, but then because they couldn't precipitate out an amine-based nitrogen salt, they assumed it was not alkaloid in nature. So it was this very important paper that is highly referenced in the pre-Mechoulam cannabinoid literature by Wood, Spivey, and Easterfield, reported in the Journal of the Chemical Society, I guess there was only one time, the one in Britain, in 1896, on the resin of Indian hemp from charas. So this is a very, very landmark study, because in this study, they went through and isolated the active ingredient for the first time and reported it in the literature. And this is an excerpt from the experimental portion. They termed it cannabinol. You could see that the formula that they prescribed to what they call toxic red oil. Toxic doesn't mean what we typically call toxic. This would mean back in the day, they would call it intoxicating. So this, they assumed, was the intoxicating fraction of cannabis, this red oil, and you'll see that they have the molecular formula C18H24O2. Well, that's not entirely correct, but given that this is, in the 1890s, it's pretty darn close. Cannabinol, you'll know, is slightly different. It has 4 less hydrogen than does THC, so it's not quite cannabinol. They were off by a little bit, but you'll have to understand that back then they were using rudimentary elemental combustion analysis, and the error that's associated with making those measurements can be considerable. Where there is no error here, though, is that they would talk about the distillation of this ruby red fraction, this red oil, which is, sounds similar to that of typical cannabis type distillate. It's got that reddish kind of color, very viscous, a viscous material, and it distills between 270 and 290 under a vacuum pressure of 15 to 35 millimeters. And then they did a redistillation at 265, 20 millimeters, and just tried to make a couple different fractions here at different temperatures to see if there was any difference. But you see here, it says when the oil cooled 60 degrees, below 60 degrees, it's set into a sticky, semi-solid, odorless mass, insoluble in water, but dissolved easily in alcohol, ether, benzene, glacial acetic acid, organic solvents in general. Sounds pretty like a THC distillate to me. We'll look at these boiling points later, and a boiling point of a pure compound is an indication of purity. And so if you're purifying THC in a distillation process, when you read the boiling point temperature and pressure, that's unique for that compound or compound mixture, whatever you're distilling. But we'll look at this data and compare this to Raphael Mechoulam's data and see if we think they distilled cannabinol as they thought, or was this actually tetrahydrocannabinol, THC. So you can see what their elemental analysis looked like. Again, pretty darn close if you consider that THC has a 80.3% carbon, 9.5% hydrogen, and here on the couple different sample trials that they run, they're getting 78.9% hydrogen and 9%. 78.9% carbon and 9% hydrogen. So pretty darn close for 1896. Remember, that's contemporary with the discovery of the light bulb. So it's pretty remarkable that they can get that close.
That's fascinating. I never noticed those ratios being so close before. I just totally overlooked that when I've read through this before. I'm glad you pointed that out.
Yeah, I think the other fascinating thing, Jason, is that that whenever you deal with cannabis distillate, it's really, really sticky. And so to me, as soon as they say sticky, semi-solid, odorless, mass insoluble in water, you know what? That sounds like distillate to me, man.
Yeah,
so
it does.
It just and there's nothing compelling that they did that would convert their extract into CBN. CBN doesn't just form by extracting cannabis and distilling it. You have to further oxidize it. So there's some more clues in some of these follow-up papers. But I think the importance of this paper is I believe that this was the first group that distilled the active ingredient of cannabis. And I don't think, unlike they believe it was CBN, I think it was actually THC. But let's look at some other papers here because some other people shed light on this. And it's just so fascinating because when you think that this is before the turn of the century, I mean, automobiles weren't on streets yet, and this work was being done. So this is just fascinating to me. Okay, so then later in 1899, so this was in 1896, this first paper. And then later in 1899, they published a follow-up paper, again, Wood, Spivey, and Easterfield from group at Cambridge in England. And you can see again, they're kind of a little off on the formula. And one of the reasons why, and I believe again, Meshulam points this out too as well, is that back in the day before they had the analytical instrumentation, the sophisticated analytical instrumentation today that we have, like HPLC and other types of chromatography and spectroscopy, where we could identify new compounds isolated from natural products. What they had to do back then was make derivatives. And they made derivatives, in this case, of their extract by essentially making a compound called oxycannabin, which is actually just nitrated CBN. So what they do is they just basically boil their extract with nitric acid, and nitric acid is a very strong acid. So it most certainly probably degrades the molecule, but it also puts nitro groups on the aromatic ring, and those nitro groups tend to be very polar, which tend to lead to more crystalline compounds. And now if they can isolate a crystalline compound, they can go back and try to figure out the molecular puzzle to figure out what the actual compound looked like.
Basically doing derivatization.
Exactly, Jason. And what they were successful at doing, and was followed up by this researcher, Robert Sidney Kahn, in 1930, was not only the identification of the oxycannabin molecule, and you could see that Kahn actually got the molecular formula correct, but they were also able to make the acetylated version of CBN, which is a crystalline substance. And so Kahn is very important, because Kahn, so if you'll notice that this goes from 1899 to 1930, well, there wasn't a whole lot done. There was a few papers, but nothing of substance. And there's a lot of these papers that are in the German literature that are either written in German, and I don't read German, but also just some of these just don't exist on the internet anymore. And so you have to, sort of piece these things together by looking at other people's papers and other people's reviews. And so what Kahn did very nicely is he published 4 back-to-back papers, one in 1930, one in 1931, one in 1932, and one in 1933, where he went through and figured out all of the loose ends of these cannabinoid structures. Kahn should be credited for basically putting together the correct formula of THC itself. And you'll see if you go through his paper here, I have a bar covering this bottom, but he references in his 1931 paper the correct formula for the first time that I see in the literature of THC, which is C21H30O2, right? So this was the correct formula, and it was proposed by Bergel, and we're going to come back to Bergel in a little bit, and we're going to come back to that paper because that's a very, very important paper. But again, this is sort of Kahn's second sort of view of, well, okay, making this crystalline acetyl derivative of cannabinol, it still was not the active ingredient that was coming from the cannabis plant. So there was still some discrepancy between what they were actually extracting and what they were derivatizing and what was actually in the cannabis plant. And so you'll recognize that in this nitration reaction, you are using strong acid. And when you use strong acid, you're probably going to scramble that double bond. and you're going to start nitrating the ring. In fact, what they call it is trinitrocannabinol, because they're able to basically do elemental analysis and show that they're putting 3 nitro groups onto some portion of the molecule. Okay, so here is the 1932 paper, and now what you see is, yeah, they have the correct molecular formula for cannabinol, which is C21H26O2. Here's the first time you see and this is in collaboration again with Burgell. These are the Burgell structures. The thing with Burgell is Burgell published in the German literature, and again, I can't get those papers, but we'll talk about Burgell in just a little bit here. But this is the first time you see in the literature in 1932, right? So this is 10 years before Adams. You see the benzopyran ring system with an amyl group. The amyl group is the N-pentyl group, the C5H11 group. You see the hydroxy group. It's implied here that this is an aromatic ring. So this would be a benzene ring. This would also be a benzene ring here with a methyl group here. So here's basically the structure of CBN in the literature in 1932. Again, that was from Burgell.
And in case anybody, do you mind going back to that slide real quick?
Sure.
Just in case anybody is slightly confused or brushing up on some of your chemistry, think about the structure that he was just pointing out with the 3 rings, that C5H11. Just imagine that as what we often call the carbon tail coming off of a lot of these, kind of traditional cannabinoids you're used to looking at. You could imagine that, coming off of there, and this might look more similar to what you may be imagining of CBN.
Well, what you'll notice, Jason, right away, and I think I pointed out on this slide, is that I got to move that bar again. Is that they have the regiochemistry wrong. So if the methyl group is here, and this is the ring junction between the methyl sort of toluol portion of the molecule of CBN, and this is the other benzene ring, then this OH should be at this carbon, and this C5 should be down at this carbon.
Yep.
So again, you have to remember that what they were doing is they were tearing these molecules apart with strong acid and then trying to figure out what the Lego pieces look like before they tore them apart. So they had this general ring structure down, which was the 2 aromatic rings with what's called a benzopyran ring system. What they didn't know was that the C5 was substituted over here and that the OH was substituted over here. That came from later work, I think, of Adams. Well, we'll see it in just a little bit here. Okay, so here was a paper that was more of a review than anything else. And I found this to be very interesting because, again, this is mostly for the British medical community. This was taken out of the British Medical Journal in 1938. So right around the time when Adams was actively exploring the active ingredients of cannabis. They point out that pioneering work by Wood, Spivey, and Easterfield, 1896. And they point out that it's unstable, readily oxidizes in air. And we know that. We know that THC will convert to cannabinol in the oxidation well, actually, what happens is the THC molecule gets oxidized to CBN, and in that process, a molecule of oxygen is reduced down to molecular water. So it needs air because the oxygen in air is the reductant, is the oxidant that is basically oxidizing the aromatic ring. And so it's interesting here that, again, what they point out is that they said the research, this would be the research of Woodsby and Easterfield, was discontinued owing to Spivey being killed by an explosion during the continuance of the investigation.
Wow.
So there's our first hash casualty there. That's our
Wow.
first person who blew themselves up. So, so yeah. So then there was this other report in the German literature in 1903 of the isolation of cannabinol. And you got to remember when they're doing these analyses, they're heating them up to do an elemental analysis. And we already know that heating up THC in the presence of air converts THC to cannabinol. And so this is just speculation from my perspective. I think that a lot of times when they were measuring cannabinol, it's only after it's been oxidized from THC to cannabinol. I don't know that for a fact because I don't have that data, but I would assume that again, if this is just extract from cannabis, there's no compelling reason on why it would actually be cannabinol and not THC. So yeah, this is just sort of like the back end of that paper, and it points out this guy Burgell, and Burgell is very, very important, and he worked with a guy named Alexander Todd, who published again some very, very important papers in this area. And so here's a picture of Alexander Robertus Todd, the Lister Institute. And this is his paper from 1939, which he co-authored with Burgell. And this is just a fascinating article that's getting more detailed on the extraction, the method. Again, once again, referencing the important work of Woodspivy and Easterfield in 1896, identifying that they made the crystalline acetate of cannabinol, which melts very sharply. It's interesting. I wonder if the crystalline acetate of CBN, so this would basically be CBN acetate, has any therapeutic value given the current interest that people have in THC acetate. The fact that it's a crystalline acetate means that it can be purified, unlike THC acetate, which I believe is a liquid, which you have to just do a distillation on. This one you could purify by crystallization. But you'll see again reference back to Kahn's important work, which basically helped clarify the observations of Woodspivy and Easterfield, and again giving that correct formula of C21H26O2. I thought this was interesting, Jason, this comment up here that marijuana has assumed dangerous proportions in America.
Yeah.
Well, you got to remember, this is a Brit over in England. So it's like, I wonder if his commentary, given the year 1939, was a little more political or, I mean, this is right at the beginning.
I was about to say, this is like what, 2 years after the Marijuana Tax Act in the United States or so?
Yeah, but at the beginning of basically World War II. So I mean,
right. Oh yeah, right. Really good point. Yeah.
You got to remember that there's probably interesting strains on the relationships across the Atlantic between all kinds of folks. But yeah, here you could actually see where they say that it was removed from the British Pharmacopoeia, again, because known as hashish, bong, charas, ganja, marijuana. I think because of it, the variability of activity, which is so interesting because that's sort of like a common theme today. I mean, people think that, how could this possibly be medicine when one plant can contain 8% THC, another plant can contain 25? I mean, that's the variability that goes along with a botanical substance, again, was kind of its undoing to be removed from the British Pharmacopoeia. But again, maybe some of the work that's going on here by Todd and others was to sort of shed light on what the active ingredient was. So again, he mentions Burgell down here again, who proposed the former structure, and that's this structure here. You can see that the OH is in the wrong position, the C5 is in the wrong position, but here's what the correct structure should look like. For a German in 1932 who was probably witnessing all kinds of crazy stuff going on in his country at the time. It's
Yeah, gee
amazing that
whiz.
this work was done at all and published in the open literature. Um,
Yeah, that
and
historical
and and
context is fascinating.
oh, it's amazing when you overlay this on what was going on in the world at the time. It's just amazing that all this was happening. So he says here that in his last communication, that cannabinol is not the pharmacologically active principal ingredient of cannabis. So they knew that they were dealing with a metastable species, that somewhere between the plant and what they were able to measure, something was happening, some transformation was happening. So here's a quote from Todd's Nature paper from 1940, talking about the resinous exudate of female flowers in a hemp plant. Um, I thought, again, this was a very interesting statement. Recently, smoking hemp under the name marijuana has attained a particular prominence in America. Well, again, enough of the commentary from the British professor here. It's like he's kind of like pointing out that this is a particular prominence in America. I wonder why, and I wonder why it's America he's concerned with and not Brits. So again, given the late date here, this is before the Pearl Harbor. So it's before the United States entered the war. Maybe he was just trying to stir things up. Who knows? Okay, so
maybe
then
he maybe he had in mind that the Americans were too high to get involved.
yeah, maybe he thought all we need to do is bomb them with cannabis and the resinous exudate of female flowers, and we'll take care of those Nazis. So this is very interesting. In his next paper, he talked about and Wood, Spivey, and Easterfield also talked about a low boiling terpene fraction, but they didn't really pay it much attention. They were more interested in the toxic red oil. But here's for the first time you see possibly myrcene and a sesquiterpene. I would bet that sesquiterpene is probably beta caryophyllene. If you go back to the Wood and Spivey data, you could actually look at the boiling point of their sesquiterpene fraction, and it is pretty spot on for beta caryophyllene. So I think that Wood, Spivey, and Easterfield data from 1896 is as solid that day that they published it as it is today.
Yep.
And it's interesting again that there's sort of like a coalescence of ideas here around myrcene, sesquiterpenes, other things that are in cannabis. Here is talking about humulene, alpha caryophyllene being isolated. I mean, they have to definitely be thinking that there's some uh, uh, constitutional makeup of these extracts that's just beyond the cannabinoid themselves. But we'll put together another class just on the terpenes in cannabis, and that'll be another fascinating deep dive that we can do. Okay, so Franz Burgell. So the only thing I can really find, I couldn't find his papers because he's co-author with Todd and a lot of the other folks that Todd was working with. And what I found that Todd wrote was a memoir after Burgell had passed away, and really pointed out, I think, the importance that he brought to that collaboration. And so, yeah, I point out here, he and Todd did some preliminary institute on the drug and showed that cannabinol, the pure compound, could be isolated with techniques available that was devoid of physiological activity, all of which appeared to reside in the intractable resin after the separation of cannabinol from the natural drug. So what I interpret that as meaning is that they isolated cannabinol, and it wasn't the thing that was that intoxicating red oil. It was not the principal active ingredient. And I think this pointed out that I think Todd knew that cannabinol is what we're isolating, but there's some other principal active ingredient that's in the intractable resin that leads to its biological activity.
And it's cool the self-awareness here, of recognizing that all of this is a result of the tools they had available at the time to study these things, and then piecing these pieces of the puzzle together to realize that, well, yeah, what they were measuring was not what was, eliciting that activity. And it's interesting to think about how this set up this sort of elusive investigation to the pharmacology of CBN in general, because that's still kind of a mystery of, like, what does CBN do in the body and under what conditions, co-administered with what cannabinoids and stuff. So on multiple levels, this is really fascinating.
Yeah, so this is what kind of kept this as an unsolved problem up to Meshulam's day. And even you'll see when we're just about to get into Adams's work, is that even Adams was somewhat perplexed, because the way that they went about isolating things often either scrambled or oxidized that ring. And at that point, you don't know what the active ingredient was, because what you're getting is some chemical derivative that was unintentionally made. So I found this to be absolutely fascinating. So this was a prelude of things to come, I call a key intermediate in the Todd synthesis. So they went and made delta 9 tetrahydrocannabinol, showed marked hashish activities in animal tests. Although the degree of activity was much less than the natural drug, at the time, Todd held the view that the active portion of cannabis resin consisted of a mixture of isomeric tetrahydrocannabinols, but owing to pressure from other work, did not pursue further study of the natural material. This indeed was vindicated some years later through the brilliant work of Meshulam and his colleague, who showed that the most active ingredient was, again, this delta 6. So the nomenclature here changed. It used to be delta 6 because of the ring number system that was associated with terpenes. When they changed the ring numbering to specific to cannabinoids, it became delta 9. So I think Todd was extremely insightful here, because I think he found that basically, no, there's a metastable compound in there. We made it. It showed marked hashish activity in animal tests. They did a test called the Gervy test. I'm not exactly sure what that is, but I think it's in rabbits. And it was just really interesting that he, without having NMR, without having a lot of the modern tools of analytical instrumentation, kind of figured this out by deduction. Here's one last comment from Todd, and again, sort of the just say no thing. So a lot of this work that was in Todd's lab was funded by Hoffmann-La Roche, a very large Swiss pharmaceutical company. And what you'll see here, I'll just skip down to this collaboration ended following the reestablishment of the controlling body of Hoffmann-La Roche in Basel. So that's after the war, because I think it was part of the assets that were seized by the Allies. I'm not exactly sure, because it's a Swiss company, but perhaps there were somehow, Hoffmann is a German name, and perhaps somehow Hoffmann was tied to that. So Roche did not follow up with these researches, not much according to Virgil, because of the negative results obtained at that point, but because Hoffmann-La Roche management did not wish to be anyway associated with cannabis, which had come to the I think this should be forefront about the time prescribed for addiction. So I just think it's very interesting that here's a large pharmaceutical company just saying no, in spite of the tremendous body of work that these folks generated under their funding. So Professor Roger Adams, there's not enough that can be said about his contribution in the pre-Meshulam cannabinoid era. Um, Roger Adams was a very famous organic professor at the University of Illinois who's the PhDs and postdocs in his group ended up becoming the organic professors that I had. The people who taught me were basically students and postdocs of people who worked under Roger Adams's umbrella. So the people who worked for Roger Adams, and then the people who worked for the people who worked for Roger Adams, and all that. So he had a tremendous impact in organic chemistry. He started a journal called Organic Synthesis, which is online and I think free, available to this day, that has a bunch of organic synthetic preps in there that are not only very useful preps, but they're checked by checkers to make sure that when you carry out a 10 kilogram synthesis of, I don't know, paranitro-aminophenol, that you actually get that compound itself. So very, very useful set of journals. And he was amazing in terms of the breadth of chemistry that he worked on beyond cannabinoids. In cannabinoids, the importance that Roger Adams can't be emphasized enough, in that he was the first one who isolated CBD from a natural variety of hemp that he found in Minnesota. He also converted CBD into THC and CBN, and talked quite a bit about their descriptive chemistries. Although the structures were incorrect, they were corrected later, 20 years later, by Meshulam. The structures were pretty darn close, kind of like horseshoes and hand grenades. You'll see the structures in a second. These, again, amazing that in the 40s he's able to basically deduce these structures at all. So he published 9 papers on his cannabinoid research in the Journal of the American Chemical Society, top chemistry journal in the world at the time. This is during beginning of World War II. So here's the full bibliography. You could see the breadth of what he was working on and the number of people in his lab that worked on these things. So the structure of cannabidiol, isolated from Minnesota wild hemp. He then published the absorption spectra of cannabidiol, then started doing some chemistry. Remember that they didn't have the sophisticated analytical instrumentation, so to determine what these compounds actually were, they actually had to do a number of different chemistries. So one of the chemistries they did is reduction. They also looked at the linkage in between the 2 rings, the positions of the double bonds and the isomers. This is, again, where they got slightly fooled. And here it is, isomerization of cannabidiol to tetrahydrocannabinol, a physiologically active product. So this is basically, again, a confirmation of what Todd had noted a few years earlier, which is that there is a more active component in cannabis, and this tetrahydrocannabinol is the physiologically active product. And also, here's the conversion of cannabidiol to cannabinol. So I wonder if the folks who wrote the Farm Bill knew that in 1940, the National Science Foundation funded this work that was published in the Journal of the American Chemical Society that basically said you can take Farm Bill cannabidiol, convert it into tetrahydrocannabinol, or convert it into cannabinol. I don't think that they contemplated this, but
I don't think so. There's a lot that they didn't really contemplate in this
right.
revision of the Farm Bill that it seems like maybe they're going to address in the next one. I wondered the same thing, because even beyond 1940, like this got reported on, like it seems like every decade, there were chemists playing around with isomerization, and figuring out how to move cannabinoids from one form to the other and things. And so that was a big gap in whoever contributed to the farm bill, that they missed that very available, freely available, open information.
Yeah, I think suffice to say, this is a very important body of work. I mean, if you look at this, it spans just 1940 to 1941, 9 papers in the Journal of the American Chemical Society. That's not a small task.
It's incredible. It's like an anthology, basically.
It truly is. It's important. Again, like I can look at this body of literature right here and totally get why Raphael came in the 60s, because there's controversy. So down here, he's talking about the positions of the double bonds and isomerization of tetrahydrocannabinols. So there's really an unfinished story here that you read. The other thing that's very interesting to note is that Roger Adams, was the first one to patent cannabinoids. He patented hexahydrocannabinol, HHC, in 1942. And that's interesting that you see HHC now sort of coming out on the hemp side of the market, because the folks who are making Delta 8 are running on to the next thing to try to make a buck. But so this work is just incredible. I've read all of these papers. In fact, some of them I've read more than once. When you read the papers, you really start to understand the person and his approach and his mind, and it's really fascinating. So here's one paper. This is from July 1941, and they didn't reproduce too well, but I hope you could see that here are so these are tetrahydrocannabinol homologs with marijuana activity. So if you look at these structures, well, now they got the phenol in the right position, because again, they did some degradation studies and showed that the phenol is in that position and the C5 is over here. Here, I blew up the structure so you could see them over here. But what you could see is it doesn't really reproduce well. The double bond is here. So this would be Delta 7 THC that he's got depicted here in 1941. And here is our friend Delta 8. So this is the first time I see Delta 8 THC in the chemical literature. This is the double bond here at the Delta 8 position. These sticks right here just imply hydrogen. So that's a hydrogen going down. There's a hydrogen coming up. There's two hydrogens here. There's two hydrogens here.
Before they cut them off.
I'm sorry?
That's before they start change the kind of typical way of drawing molecules, and they just eliminated those
right,
sticks altogether.
Where it's just implicit that there's two hydrogens there. If you look at these structures, though, this is amazing, because this is 23 years before Meshulam's paper, and Adams is basically there. And he's standing on the shoulders of Burgell, who is standing on the shoulders of Kahn, who is standing on the shoulders of Wood, Spivey, and Easterfield. So it's this continuum. And I think that, again, Roger Adams had to know that prior work in order to do new work at the frontier. So you're looking at the frontier in 1941 right here. Okay. In 1942, this guy, Woolner, this is very interesting. He's from the U.S. Bureau of Narcotics Laboratory, which is very interesting. This is basically the precursor to the DEA. And so the government had set up a Bureau of Narcotics, and Woolner was basically the chemist there. and he did some very, very important work and is a very careful chemist. And I've read several of his papers. And again, just think that he's one of those people that we need to pay attention to. So this is a paper from 1942. And what he describes here, you could see that he's taking charas of Indian origin, so probably indica, from the fall of 1939, and alcohol extraction in the cold yielded a viscous brown oil. Well, that sounds about right, right? Um, and then he talks about these grayish sandy powder. These are probably the fats and waxes again that sort of precipitate out of the cold ethanol. So then he basically does a crude distillation to basically make a distillate. So, you know, there's no real reason why this is going to be converted in from, depending on the nature of the charas. If the charas is enriched with THC, this process right here is probably just going to give us a THC distillate. He acetylates the crude distillate, right? And remember, the acetyl group on CBN makes a crystalline product, but here he's getting a cannabinol acetate that did not crystallize. Gee, I wonder why it did not crystallize. Because it was the acetate of THC, not the acetate of cannabinol at all. So he then, this is very interesting. This is the first, I wouldn't say the first, but this is very early column chromatography. You know, column chromatography was really developed in the 60s and the 70s when macroporous silica became available. But what you see here is they're taking a grade A activated alumina, and they're mixing it with this high flow supercell, which is probably some just filtration aid like sea light or something like that, creating an absorbent material like that, and then passing the stuff through to basically try to get some purification. Well, they could also purify it under high vacuum too as well. And what you can see here is, again, very nice data that shows not only the specific rotation of what they're getting out. The higher the specific rotation, the more likely it's a optically active product that you're getting from there. But they're also getting pretty good analytical data that talks about the composition itself. So here's THC acetate. You know, there's a lot of interest again with these hemp folks who are basically making now THC acetate derived from their Delta 8. Again, just to move on to a different derivative. Here it was shown back in 1942 that those compounds can be made.
And it's confusing on the market too. They just keep saying THCO or THCO acetate, and they're not differentiating between Delta 8 or Delta 9.
No, they don't. Well,
yeah.
what I can tell you, Jason, and I've done a little bit of detective work, is most of the stuff I see out there is Delta 8, because Delta 9 right now is a tightly regulated, in most cases in the States, seed to sale regulated, and it's difficult to get stuff out of that market. Yeah. In California and Oregon, there is some leakage between sort of like the legacy market and the legal markets, but it's still something that gets kind of muddied over, because the point of where these things are sold are in some of the most cannabis unfriendly states, and people just want this stuff because they want to get
not go to jail
high.
And pass
They want the psychoactivity. And if you think about it from that perspective, this is only made possible through the ridiculousness of prohibition itself.
Yep.
So this is ridiculous. So then this is fascinating. So what he did is he took again, this is Wallner. He took his tetrahydrocannabinol acetate and now did a dehydrogenation. So now he's removing hydrogen to basically turn it into CBN. And now he should get a crystalline acetate, and lo and behold, he does. The yield of cannabinol acetate was this, and it was indeed a crystalline solid. So it's very interesting because what you see here, and also he hydrogenated it too as well. So, I mean, this is basically HHC acetate being made by our friends at what became the DAA. Okay, now enter Meshulam. So this is the most important paper that we're going to talk about today because in this paper here, this was Raphael's, I think, second paper. He published a communication before this, but this was the first paper where this data occurred in 1964. And so what he did was a chromatography of a hex strain extract of hashish, which then he repeated. He first did it on fluorosil, then repeated it on alumina. And that way he could separate out the inactive, what was thought the inactive cannabinol from the tetrahydrocannabinol. It's interesting at this point because if this hashish is like most hashish, it's not tetrahydrocannabinol, but it's actually tetrahydrocannabinolic acid. But they just didn't know that at the time because when they were doing these studies, they were doing distillation of the products, and if you distill a cannabinoid acid, you end up getting the decarboxylated material. So they assumed that it was tetrahydrocannabinol, but in essence, this extract was probably enriched with tetrahydrocannabinolic acid. So then it was a very common practice to take compounds and convert them into a crystalline derivative that can then be purified by crystallization. And so what Raffi took advantage of was the fact that tetrahydrocannabinol had an OH group, and the OH group is something that can react with a compound called an isocyanate. In this case, it's the 3,5-dinitrophenyl isocyanate. And so when you react this phenol with that isocyanate, you form a crystalline derivative, which can be purified by crystallization and characterized by the melting point. So it's similar to the boiling point of a pure compound. The melting point of a pure compound is an indication that you have a pure compound. He also took an optical rotation to make sure that it was optically active and that his preparation up to this point didn't somehow ruin the optical activity. He did an analytical analysis and also got very close to where it should be based on the empirical formula. So then what he does is he takes that material and does a mild basic hydrolysis with a little bit of base. And what that does is it removes the nitrophenylurethane group, which is extracted into the water layer, which goes into the organic layer, is now the pure compound. So now after that hydrolysis, he got the pure compound, which then he did a distillation of, and it boiled at 155 to 157 centigrade at a vacuum pressure of 0.05 millimeters. It's very important to understand that the boiling point of a compound measured under a vacuum is done so to lower the boiling point of the compound. So when you distill something under a vacuum of 0.05 vacuum pressure, you're needing less heat to boil that compound. And so it's often reported that the boiling point of THC is 155 to 157, but they don't include that that's at a vacuum pressure of 0.05. If you try boiling THC at 155 to 57, which is about 315 degrees Fahrenheit, it's not going to boil very readily because you're not at the boiling point. That's the boiling point under a vacuum. And so the boiling point of THC is most oftenly reported as these numbers, which is correct, but at a vacuum pressure of that. So it's very important to know that that's how the vacuum, or that's how the boiling point was measured. It was measured in a short path distillation apparatus, I'm sure, and he's able to get a very, very good, accurate reading of the boiling point. Then to characterize the material, he used a new technique called proton NMR. So when you see a 1 next to an H, that's a chemist's abbreviation of a proton. So this is proton NMR. NMR stands for nuclear magnetic resonance. It was a new technique that was developed in the 50s and later in the early 60s by notables like Jack Roberts and others to characterize organic compounds, because each proton in the molecule has a different resonance frequency or energy, I guess you'd say, in a magnetic field. And so where those occur is indicative of the electronics in and around those protons. And so this was a new technique that wasn't available to Adams or Wood, Spivey, and Easterfield or Kahn or any of their predecessors. So Raphael used this technique to properly determine where the double bond was in the molecule. And this signal right here at 6.35 is very important. I'll show you an NMR spectrum later where we will see that proton. But that proton turns out to be this proton here at what used to be called C2. This is no longer C2. This is now carbon 9, I believe, in new structure. But in the old nomenclature, this was C2. And what you see here is that proton at C2 is 6.35. The aromatics are at 6.18 and 6.0. Those are these aromatic protons. And so he was able to basically characterize this compound versus the delta 8, which was previously characterized by other folks. Okay, so let's look at that boiling point. So his boiling point of 155 at 0.05 vacuum pressure, when you put into this relationship in the nomograph, a nomograph is a tool that's used to relate measured boiling points at a particular vacuum pressure to the corrected boiling point at standard atmospheric pressure. And so what you see is if you plot Mechoulam's data, which is 0.05 vacuum pressure with 155, it crosses this line at about 425. And if you look at the Wood, Spivey, and Easterfield data, they had a vacuum pressure of 40 and had a boiling point, 46, and had a boiling point of about 300. It crosses the line at just about the same spot. And here's a measurement that I made a few years back where I had a vacuum pressure of 0.3. And my boiling point that I was reading was 184. All of these numbers cross this point at the same line, or at the same point, I should say, which basically indicates that THC's boiling point at atmospheric pressure is not 155 like it's often reported, which is 315 Fahrenheit. THC's boiling point is 425 degrees centigrade at atmospheric pressure, which is about 797 degrees Fahrenheit. So there's an online version of the nomograph that's been made available by our friends at Sigma Aldrich. So if you go to this website, what you can do is you can plug in uh, Meshulam's numbers of 155 down here. That's what he measured, and a zero point zero 0.05 vacuum pressure. And what you'll see is that the corrected boiling point is indeed about 425 degrees centigrade. So, if nothing more from this class today, you've learned the correct boiling point of THC. That's good.
And that's been repeated incorrectly. I mean, I taught it incorrectly for a while because it's been repeated in literature so many times without that nuance around the vacuums of some of the early measurements.
The vacuum pressure, right?
Yeah.
Yeah. I mean, I'm fine if they have a little asterisk and it says at a vacuum pressure of.
Right.
But when they say that the boiling point of THC is 155, now you have to understand too, Jason, that you can evaporate a bowl of water in a room without that room ever getting to the boiling point of water.
Right.
Right.
Yep.
So what it depends on is what the saturation of the air is in that room. And that bowl of water is going to disappear a whole lot quicker in California and Colorado than it would, say, in Florida.
Yeah.
Just because of the overall relative humidity in the room. So there's a lot of things that go into whether something volatilizes, and it doesn't necessarily have to be heated to its boiling point, but when they do report boiling points, it often irritates me that they'll say, oh yeah, the boiling point of THC is 155. And then there's no footnote to the Meshulam literature that, well, it's 155, but that's at a vacuum pressure of 0.05. So I always like to clarify that because, again, I think that's something that's out there quite a bit. And like you said, it's something that unless people know or understand sort of the basis of it, they'll probably continue to think that way.
Well, and it's great too that you pointed out these tools that people don't have to take your word for it. They can literally go in and measure this themselves and compare and get to these same points. It's always great to have
that's right.
That empowerment.
So what you can do is if you're running a distillation, and I've talked with a lot of people who make distillate, and they're like, yeah, I don't really take a pressure or temperature reading. I'm like, well, that can tell you what the makeup of your distillate is, and when they understand that, then they start paying attention to it. So, like if you're distilling hemp and it's got CBD, so CBD's boiling point is about 5 or 10 degrees higher than THC's. So you'll see a different number. Yeah. And it's interesting because you could actually see fractionation and you could change your collection accordingly. So maybe what you can do in the front end of your distillation is kind of remove the THC rich part of that hemp fraction. And maybe on the back end, you change your collection flask and now you can collect a more CBD lean fraction because you've co-distilled out the THC. The boiling points are very, very close. And even the boiling point of CBN is close, but the boiling point of CBN is significantly, it's different. So I think you can argue that, again, we know that Meshulam's data, if you look at it, I mean, there's no way. So Meshulam definitely isolated Delta 9 THC. There's no question about it. But it's very peculiar that if Wood, Spivey, and Easterfield supposedly isolated cannabinol, that it looks like it goes through the same point. And that can't just be a happy accident. I think that Wood, Spivey, and Easterfield, just like Walner, what they actually isolated wasn't cannabinol at all. It was THC. But, again, I'm speculating 100 years later. So, it's, It's interesting. So Raphael, again, very important researcher in cannabis chemistry. I think part of the reason why is because he was over in Israel and can do this work, and it didn't have the negative connotation or the negative stigma that it sort of had here in the United States. In fact, he tells the story that he wrote to the National Institutes of Health looking for money, and they turned him down, saying that there was no interest at the time. This would be the early 60s in cannabis chemistry at the National Institute of Health. Well, after he published his paper in 1964, he got a visit from the head of the NIH, who I believe smuggled 10 grams of THC back to the NIH lab in Bethesda and ended up doing a bunch of secret US government research on it. And I believe after that, Raphael started getting a never ending flow of money from the NIH, because I think the NIH, after his initial discovery, recognized that this is extremely important research to be done. So here is a paper after 1964. So this is in 66. I love it that he just titled it hashish. I mean, come on now. This is in Tetrahedron, which is a hardcore chemistry journal based in England, Elsevier Publications. They are purist chemists. So to title the paper hashish, paper number 7, the isomerization of cannabidiol to tetrahydrocannabinols, I think was very, very bold of Raphael to do that. It
gets people's attention for sure.
It certainly does. And what you can see here is he talks about the inactive cannabidiol as acids. So this is the first time you start hearing about cannabinoid acids. And to be either in the delta 1 configured, which is our delta 9, or a mixture of 2, which is the delta 8, right, which is the delta 2, right? So here, that's what we're looking at right here. So here's delta 8, here's delta 9, right? Here's delta 9 as the acid. So this is the first time you see the acid. And what you could see here is he hydrogenated, not the acid, he hydrogenated the decarboxylated material to make HHC as a mixture of beta and alpha anomers here at this position. But he did not hydrogenate THCA, and that's what our invention is. We hydrogenate.
That was like the big thing that you kind of noticed and wanted to explore.
Well, yeah. Well, we found basically a hole in that literature, and I think the way I put it on LinkedIn, Jason, was I patented it so big pharma couldn't. So, yeah. So here's a hydrogenation of delta 8. So this is kind of what those hemp guys are doing out there. They're hydrogenating delta 8. You could hydrogenate delta 9. You could still make HHC. He recognized, again, the beginning of the understanding of the biosynthetic pathway, which is that geranyl phosphate reacts with allylic acid to make CBGA. That CBGA then undergoes a oxidative cyclization to make CBD. It's thought of that CBD was the precursor to the tetrahydrocannabinols, but it was later shown that a different synthase operates on CBGA to convert it into the tetrahydrocannabinols. But you can see here, he's also pointing out this is delta 7 CBD, and hydrogenated CBD here. Here's a ethoxylated HHC compound and some other derivatives. But this is, again, where you find the understanding that delta 9 converts into delta 8, and it's a one-way trip. See how this arrow just goes one way? It goes one way because this is thermodynamically downhill. And if you look at this, he says there's a shift in the olefinic proton from 378 cycles per second. Well, he was doing this at 60 megahertz. So when you divide that by 60 megahertz, that's 6.3 to, let's see, so I want to divide 322 divided by 60 goes to 5.3. So there's a 1 part per million shift in this vinylogous proton in going from this structure to this structure. And that was observed by Scialdone in 1968. Here's, again, a little bit more chemistry that talks about the conversion to CBN. He's using chloranil here, which is a very potent antioxidant. It's not anything I would recommend for anything for human consumption, but certainly an antioxidant or a quinone oxidant that can basically take THC and convert it into CBN. He found also that, again, you make these isoderivatives. So in this case, this is a compound called cannabitracin. And then this compound down here is one of the iso compounds, which is one of the byproducts that's formed. We'll talk a little bit more about this in the conversion of cannabidiol over its delta 8. Okay, now comes Raj Razdan. And so Raj Razdan, who's in Cambridge, Massachusetts at the Arthur Little Company, did some very nice work to look at the isomerization of, this was, again, delta 9 THC. This is the old nomenclature. The active component constituent of hashish, completely isomerized into, this would be delta 8, when refluxed in paratoluene sulfonic acid and benzene. So what's happening here is the conversion of CBD by protonation of the double bond and closure of this ring to form the benzopyran ring structure. And what you see is that the double bond does an isomerization from the delta 9 configured material over here to delta 8. And they're also showing that happening with a Lewis acid, boron tribromide, and here starting with delta 9, converting it again just with paratoluene sulfonic acid. So what's going on here? Well, why is this chemistry? Is it something that again is leading to a particular set of intermediates, or are there sort of a way that you can funnel everything into, say, one compound? Because this is kind of the crux of the matter right now, because people are starting with, in this case, they're starting with this isomer of CBD8, and they could make a variety of different compounds. They could make these iso compounds. They could make these ring closed compounds. And so what is being made? Well, Razdan followed up with a very elegant paper that dealt with how he was actually going to make these compounds from scratch. And that is by condensing this compound, this mentha 2,8-diene-ol with olivitol. And so in this reaction, what's happening is you're taking this terpene precursor, reacting it with olivitol at one of the unsubstituted positions of olivitol. And you'll recognize that this compound here is basically CBD, because it's reacting at the position in between the two hydroxyl groups of olivitol. However, there's another position it could react at. Rather than reacting here at the position between the two hydroxyls, it could react at this other position, which creates an abnormal isomer. And this shows the same series with delta 8 THC being formed, and here the abnormal version of delta 8 being formed. So the abnormal version, you'll notice the normal version has the C5 tail on this carbon, but the abnormal version has the C5 tail here. That's the two differences in these structures.
And I want to point out to our students too, because I mentioned briefly abnormal CBD when we were talking about the endocannabinoid dome and some of the research that's been done on that. Keep this in mind, because this concept of these abnormal cannabinoid isomers will come up again later.
Yeah, so the abnormal isomers were problematic from the point of view to, like, if you're starting with CBD and you're treating with a Lewis acid, you're basically going to scramble the CBD to the abnormal substitution pattern. And the thing with the abnormal substitution pattern is that there's two positions here, but there's only one position here. So statistically, if this is all equal, you're going to get 1 to 1 to 1. So that means you'll get twice as much of the abnormal isomer than you will of the normal isomer. So this is extremely important, because if you're starting with CBD and you're treating with a Lewis acid, it can literally tear apart the molecule and recombine in an abnormal way. Even if it doesn't recombine in an abnormal way, if you take CBD, when you do the ring closure with BF3, you end up getting this, which is called an iso product, and you end up transforming that iso product into an isomeric iso product. So this is called iso 8. This is called iso 4-8. And the iso 8 and iso 4-8 pairs are also in the abnormal series as well. So now you can see, Jason, when you're trying to make delta 8, this is the only delta 8, yet you could have one delta 9, you could have iso 8, iso 4-8, abnormal 9, abnormal 8, abnormal iso 4-8, abnormal iso 8. So these are all the particular products that you could potentially make. And it's interesting to note, too, that they all have the same molecular weight. So they're all isomers of each other.
Yep.
So when they're all isomers of each other, they're all going to have molecular weight 314. So it's not like you can use the mass spec to figure out exactly what's going on.
And to just highlight that, and I'm sure we'll talk more about the Delta 8 issue probably towards the end too, but this creates, depending on how an analytical lab is set up and prepared to look for some of these things, it's very easy for all of this to look like one thing. And so sometimes you can get analytical results that may say something is quite pure in Delta 8, but there can actually be quite a number of other molecules that are just so similar in their size, their weight,
They co-elute on the HPLC.
They can.
What we heard the other day, so we were on a Delta 8 call with our journal club with the American Chemical Society Cannabis subdivision. And on that call, there was an expert from KCA Labs who was saying just that, Jason, is that some of these products, which have the same molecular weight, co-elute with the Delta 8 and Delta 9 and shoulder those peaks. So they know that there's something underneath the peak that kind of distorts the peak a little bit, because those peaks should be very nice, sharp, isosceles triangles, right? They shouldn't have kind of like a shoulder to them or anything.
Absolutely.
So there's a lot of products that could potentially form from that. And we're actually going to take a little bit deeper dive into that conversion chemistry toward the end. Okay, so what's happening here is very interesting. Here on the left, what you see is loss of CBD with time in the reaction with boron trifluoride etherate in a dichloromethane solution at 0 to 0.5. So what you'll see is in a first order way, CBD is consumed, and in this case, delta 1, which is delta 9 THC, is produced. So boron trifluoride does convert CBD, which goes down in concentration, to THC delta 9. But as you can see, from the very beginning of the reaction, you are forming the ISO 8 product. Not ISO 48, but ISO 8. So ISO 8 forms from the very beginning. You can't keep it from happening. So here at this time point here, you already have about 10% ISO in there. Even though you have about 40% delta 9
Wow.
THC, you still have 10% of something in there that doesn't form. So the isomer does not form in cannabis. So I'll go back to the previous chart. So that would be this isomer right here. So this isomer is forming concomitant with this delta 9 THC, and you kind of can't keep it from happening. Okay, so now on this side, what they're looking at are the compositional changes in the reaction of abnormal cannabidiol catalyzed by boron trifluoride, again in the dichloromethane solution at 0 degrees. So here, loss of abnormal CBD is giving rise to abnormal ISO, abnormal delta 1, which is delta 9, and abnormal delta 9 THC. So these are the kinds of products that, again, well, this product is in cannabis, but these two products aren't. This product is not. So now when you look at these chemical conversions don't have versions that happen in the plant. So the plant is carrying out this chemistry not with boron trifluoride, but it's carrying out this chemistry with enzymes that sterically arrange the molecules in favorable conformation to inhibit the formation of these abnormal and isoisomers. Okay, what you're looking at here, this is very interesting. So this is the compositional changes in the reaction of CBD with para-toluene sulfonic acid. So now you see that iso formation is delayed till about 40 minutes into the reaction, you start to see isos being formed. But before that, you only see delta 9 THC being formed. And at the point that you start to see isos being formed, some of that delta 9 starts to now convert over into delta 8. Remember, this is delta 8, not delta 1,6. So you start to see delta 8 being formed. You start to see isos being formed. But early in this reaction profile, you see very cleanly only the delta 9. And what this speaks to is the subtlety of these reactions. And the subtleties of these reactions is that when you use one Lewis acid, you see behavior like this. When you see another Lewis acid, you see behavior like this. And so there's a lot of compositional space that's in between that can be explored. And so people did indeed pick up on that. And so here's Meshulam now in 2002. So we're moving up in time considerably. And in 2002, this was a review that was in Chemistry and the Physics of Lipids. And what you can see here is that Meshulam, again, goes through this scheme of starting with cannabidiol, treating with, in this case, either BF3, Lewis acid, or ethanol, which in HCl, which is a protic acid, and converting CBD into delta 9 THC. Well, here he's got an arrow that shows para-toluene sulfonic acid goes to delta 8. And here an arrow that says sulfuric acid with methanol goes to this methoxy ether. So what happens is you protonate the double bond, and then the alcohol just intercepts that carbonium ion. So you end up incorporating a methoxy group from the methyl alcohol, but ultimately this undergoes acid catalyzed elimination to basically make delta 8 THC. So there's a lot of things going on here, and I think Mechoulam recognized it and sort of encapsulated it in this somewhat confusing slide because it's hard to tell exactly where things go. It looks like no matter what direction you go, you end up with delta 8, right? So in this case, he was able to show that you could hydrogenate selectively this exocyclic double bond, and now you can't close the ring system here to make the benzopyran ring. You could only close to make the iso compound. So they made the iso compound with BF3, which they can also make after they hydrogenate iso that's made by treating this with BF3. So a little confusing, but basically what it says is that you do get a bunch of different things out of this conversion of CBD with acid. So along comes this group in 2020, and this group of Italian authors, Marzullo and company. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Well, I wish I had 8 colleagues to work with. I could probably publish big papers like this too. But this is a really very, very interesting paper because what they talk about is exactly the chemistry that we've been talking about, which is this CBD chemistry and this conversion to either delta 9 THC or delta 8 THC or these iso compounds like we just talked about. So there's the iso 4-8 and the iso 8. And so what determines what's in these mixtures in this acid catalyzed intramolecular cyclization? So let's take a little bit of a look at this. So in this case, what you're doing in path A, you're protonating the double bond on the methyl cyclohexene ring, which is then trapped by the phenolic oxygen up top to make the iso ring structure. And then iso 8, which is initially formed, then isomerizes in the iso 4-8. You'll see that this double bond just isomerizes. So it's a tetrasubstituted double bond, and that's the thermodynamic resting point for this. So path B, what you're doing is you're protonating this exocyclic double bond, generating this tertiary carbonium ion, which is then closing on the ether or the phenol oxygen to make the ether linkage to give first the kinetic product as delta 9, and then under thermodynamic conditions, isomerization of the double bond from the 9 to the 8 position. So in this chemistry, you're starting with a pure compound, and you're basically opening up the gate to 4 different other compounds. So let's look at what kind of conditions and what kind of temperatures and times give what. So BF3 is a very commonly used Lewis acid. TMS triflate is a more specialty Lewis acid. Indium triflates recognize that indium rare earth metals are indeed Lewis acids. Zinc bromide and titanium chloride are just other common Lewis acids. These solvents like dichloromethane or toluene or acetonitrile are common solvents. And here are the reaction conditions. And what you can see is that at a variety of different conditions, you get a variety of different products. Here's delta 9, here's delta 8, here's iso 8, here's iso 4-8. So what you can see in terms of getting a reaction exclusively to make delta 9 is that the best one they have looks to be this one right here. Indium triflate at minus 10 degrees for 6 hours gives them a 52% composition that contains 52% delta 9, 6% delta 8, and 4% iso. Still, it has a fair amount of other stuff in there, But what you can see is that as soon as you warm that reaction up to room temperature, that delta 9 converts all over here into delta 8. So you see basically what I call driving off the delta 8 cliff is somewhere between minus 10 at 6 hours to room temperature at 48 hours. It goes from selective for delta 9 to selective for delta 8. And you can see you can easily make selective for delta 8. Here's 98% delta 8. This is just toluene, 0 degrees, 24 hours. Here's 93% delta 8. That's TMS triflate, trimethylsilyl triflate to strong Lewis acid. It's interesting how boron trifluoride doesn't really give as high a yield as some of these more specialty Lewis acids. Okay, so here's some reactions where they screened different acids, different protic acids. Protic acids, unlike BF3, which is an aprotic acid, right? These don't have protons. These just have a Lewis acid metal. Here, the proton itself is the Lewis acid. So what you can see is that you can make delta 9. You can make delta 9 in 94% with paratoluene sulfonic acid and dichloromethane at 36 hours. And you can't really see it. Well, here's selective for delta 9, but you still have 11% delta 8. So 48 hours toluene, room temperature, paratoluene sulfonic acid, they're seeing 82% delta 9. Very interesting. And here with these acids, you see no reaction. NR means no reaction. And it's interesting because I know that there are players out there who claim that you could just use acetic acid.
That's what I was going to say. Yep, there's some common like recipes online about using acetic acid or sulfuric acid.
Yeah, and again, Jason, maybe under these conditions, acetic acid is no reaction. So maybe if they heated it up,
right,
acetic acid would be strong because you're putting more energy into the system. But they're not heating it up. This is at 0 room temperature or minus or whatever. I mean, these are pretty mild reaction conditions. But just to see no reaction there is pretty interesting.
Yeah, and this really highlights, these important nuances around, what you're using to drive these reactions and why the concern around delta 8 is, what is the knowledge level of the chemist actually doing these reactions? Do they understand, what they're doing, or are they following a recipe they found, on a forum online? And that's kind of the big.
Yeah, I would say that this one looks pretty promising. I mean, here's catalytic amount of toluene sulfonic acid, so that's 10 mole percent, which is good. 96 hours, and they get a 9% mixture of THC and 89 of delta 8. So maybe there's some way of converting this 9 over to delta 8, and there's no ISO. So that's very interesting. No ISO, that's good, because up here in the boron trifluoride, whoa, we're seeing lots of ISO. So they don't see ISO with the paratoluene sulfonic acid under these conditions. So that's good. But here's paratoluene sulfonic acid and with hexane, and you see 13%. Now I think this is more than a catalytic amount. I think what's key here is that this is 10 mole percent. So this is a catalytic amount. You should only need a catalytic amount, but do all the hemp hustlers know what a catalytic amount is, or do they just dump in a bunch until it's done? So yeah, it's interesting to think that, again, you have possibly the lead to a good route to make a single compound here, but I think you'd want to, develop a process. Obviously, if you're in a hemp market, you can't be selling delta 9. So you'd have to convert all of this delta 9 over here to delta 8. And if this delta 8 is free of the acid catalyst and is free of other iso components or the abnormal components, maybe what we're looking at with this particular example, Jason, is a potential way to make a clean Delta 8 product. So I'm sure the Delta 8 producers, some of them know about this study and some of them know about this paper. And maybe when this paper came out in 2020, some of the work that's now going on in Delta 8 is kind of like looking at, conditions that are giving exclusively these kinds of things. But when we talked to the guy at KCA Labs last week on the Delta 8 Journal Club call, he said that all of the products that they've looked at, all without exception, have these ISOs and other products in there. So they haven't seen a clean Delta 8 sample, but maybe again, this entry in the table gives some hope that there could potentially be a clean sample. I have nothing against Delta 8 THC.
Mm-hmm.
I just have something against people who are making a product with all kinds of unknowns in there and just putting it out there to make a buck. I think if they developed a commercial process that looked like this and didn't have this Delta 9, but had a lot of Delta 8 in it and none of the ISOs in it, I think that could be a viable candidate for commercial production, provided that the product that's produced is within the commercial specifications.
That's the big thing, like having that quality control element in there to be able to verify that, and then that ties into the issues around the analytical testing and other issues that we're getting close to ironing those things out, where I do think there could be, depending on the legality side, and kind of what states and the feds kind of do with this, but there is a route here to get really great pure Delta 8 THC. And like you said, I mean, there's been tons of research on Delta 8 THC. As a molecule, I don't think there's anything to really worry about with it. But yeah, it's all of these little things. And to echo, because I didn't make it to that ACS meeting, I wanted to, but my experience is the same. I mean, I haven't tested a lot of these newer products that are on the market, but from the work that I was doing back from like 2014 to 2018, 2019 or so, we were seeing lots of Delta 8 products even then. And it really was producer specific. So out here in Southern Oregon, we have, I don't know if you know this or not, there's like an extremely high concentration of chemists in Southern Oregon for different reasons. But there were one or two producers that seemed like they understood what they were doing. They understood synthetic chemistry, and they could pull off what seemed like, as well as we could figure out based on the tools we had available, because there weren't reference standards readily available for a lot of these ISOs, and then there's like XO-THC and stuff too. That was hard to tease out, but the best we could figure, like there were one or two, but I mean, that's out of a lot of samples. So yeah, the vast majority, I'd say 95% or more, carry these other compounds in them.
Well, the other thing too, I mean, this is one of the things that people always talk about is, again, you mentioned the analytical standards, and the analytical standards are good, and you have to be able to get baseline resolution between your impurities and your product. So if you have analytical standards that basically show that you have something that coelutes, I think that's what I heard the other day from the folks at KCA Labs, which is that these things coelute on the HPLC, and you can't really change the column or the conditions enough to get them to not coelute.
Not for a commercial lab, because the run times and everything get absurd when you try to do that.
What they did, Jason, which was the absolute right thing to do, is they did a separation by GC instead. Mm-hmm. And now, recognizing that, again, everything is going to be decarboxylated anyway, so it's okay to do the analysis by GC, but they could actually get, and they only know this because they made the standards. They had the standards made. So they made the ISO 4 and the ISO 48. They had to get those made. And when they found that they co-eluted, and I think that the way they did that is they just spiked their samples
Mm-hmm.
and they saw that the peak went up. And that's a way of indicating that when you add that sample, that standard sample to that thing, that you're basically seeing something that's underneath that peak. Well, in the GC, they could actually resolve those compounds. They could actually see different peaks.
Yeah.
So they were able to separate out the isos only by GC and not by HPLC. But now when they correlate their GC results back to the HPLCs, they can basically say, yeah, there's ISO and iso-48 in all of those samples. So so two things. We need companies to make those standards because it's probably expensive to make them.
Yep.
And we need to set some type of product specification on those processes based on the safety of those unknown compounds, because it's not that they're not unknown, but the plant doesn't make them. So if you're just going to say, well, it's cannabis derived, well, it is cannabis derived, but the plant doesn't make the isoisomers. So what type of impact does that have on human safety? Right? Like you can't
Right. Yep.
you can't just say, okay, I have 10% of the ISO 4 in there, and we're just going to let it out as a vape pen or as a whatever. Well, the safety of those things in humans is unknown. And so I think what's important here, and the reason why I like this slide, is because this shows that there is a set of chemistries that can avoid the ISO. So here, delta 9 is naturally occurring. Delta 8 doesn't naturally occur, but it's kind of like a cousin of delta 9, so we'll give it a pass. So if you could just convert this delta 9 over here to the delta 8, again, there might be a process here that doesn't make the ISOs. They did not actually specifically look for the abnormal ones. And when I asked the KCA lab guy about the abnormal ones, what his quote was back to me is that we do see other peaks that have the same molecular weight, and certainly those certainly would qualify as that. So I guess to sum up this area is that this conversion is out there. It's happening. People are doing this. What you can see here, this is again from the paper from the Italians to basically separated out their delta 9 and their delta 8. They're able to make the NMR assignments. They did the same thing on the ISO compounds. And this is wonderful for people like me, right? So this is my crystal ball. My crystal ball is that I can look in the literature and I could say, okay, here's what ISO 8 looks like. Here's what ISO 48 looks like. What you'll see here noticeably, and this I do believe is incorrect, this should be a doublet at 5, not a multiplet. This should be a nice doublet. And what this doublet corresponds to is position 9, which is this exocyclic methylene. And so when this methylene migrates into the tetrasubstitute position, basically that peak at 5 goes away. There is no peak at 5 anymore. But this peak at 5 is a nice doublet, and I'll show you in an NMR that I ran what that looks like. So this is their NMR, and what you can see in their NMR, if you look at 5, that doesn't look like a multiplet to me. That looks like a doublet. So this doublet right here corresponds to that methylene at the end of the molecule. And what you see here is here's the delta 8 peak. That's that peak at 5.3. Here's the 6.3 vinylogous proton, right? That's that big shift in 1 ppm that we see. So here's delta 9 and here's delta 8. And what you can see here is their spectra isn't very well resolved. And when I put these slides together, I said, well, I got a much better resolved spectra. So this is actually a spectra from my lab. And what you can see here is here's the 3 delta 9 peaks. Here's the vinylogous peak, and here's the 2 proton peaks. And what you see here is the delta 8 peak, and you see another 2 sets of peaks that correspond to aromatic protons. Those are basically the delta 8 isomers, and these are the phenols. And what you see right here at 5 is that nice doublet. That doublet at 5 corresponds to the ISO 8 compound. And here's some of the other mixtures that are in there. So again, this is a reaction of CBD with less than equivalent of boron trifluoride and dichloromethane at 0 degrees for 1 hour. And this is what you get, which basically shows exactly what Razdan observed, is that when boron trifluoride is used, you generate about 10% of the iso compound. You generate a lot of delta 9, but you do see some leakage over into delta 8. So this group came out with this article, and this is really a tour de force of everything you're finding out there that can be made from CBD, including CBN and THC. And so here's their table of compounds. And so overview of products formed upon acid catalyzed degradation of CBD. So there's a number of different compounds, notwithstanding delta 9, delta 8, delta 10, delta 11, hydroxycannabidiol, dihydroxycannabidiol, hydroxy and methoxy hexahydrocannabinols, methoxyhexahydrocannabinols, the iso compound, iso THC. So, this is this is what's out there right now. What's out there right now in an unregulated market. So you could see probably why it's so alarming to some people. Okay, so I'm going to finish up by just giving a little bit of perspective here that I think will be somewhat enlightening, and hope I'll open up the floor for further discussion. And that is that, cannabinoid chemistry has come a long, long way. And this report right here from Hanus, who is one of Meshulam's co-workers, is available online. It's a wonderful, wonderful review that talks about just about every piece of cannabinoid molecules research that's out there. It just includes biosynthesis, doesn't really address too much on terpenes, but talks a lot about cannabinoids and cannabinoid derivatives. Excellent, excellent article.
And anything from Appendino too. It seems to always be really forward-looking.
And here's the man himself. Raffi did his own deep dive in chemistry. When he, first of all, he should be wearing some safety glasses here and probably some gloves, I would imagine, too.
I was about to say, maybe he's just gotten to that point in life.
I think he was just posing for a picture here.
Yeah.
But he's really done an amazing body of work, and his deep dive into cannabis chemistry was this article. I believe this was published in 1967, and he actually goes into a pretty decent review of the literature in this area, and it's just fascinating what was known up to that point. So this is, I would say, probably Raffi's own deep dive into cannabinoid history. And then later they published a review in 2000. So this is already, 21 years old. Hard to believe 21 years has passed.
Yeah, it's crazy.
And this is just a fascinating paper. It really is. It's a wonderful historical overview of cannabis. It touches on the Adams work. It touches on the Kahn and the Woodspivy and Easterfield. They even go into talking about Schlesinger and decorative. So this is also a very excellent review of pre-Meshulam cannabis chemistry as well. His idea back 21 years ago was the future directions. Where will the field go? And I love this ancient Jewish saying that says that prophecy powers are given to fools only, with the hope that we shall not be exposed to fools who believe that chemical research in the next decade will proceed in part, at least along the following paths: identification of novel receptor sites and ligands, the elucidation of three-dimensional
Mm-hmm.
structures of plant, synthetic, and endocannabinoids for binding receptor studies. Some of that work has actually been done and published. Novel chemical approaches to specific drugs against pain and inflammation, multiple sclerosis, brain damage, etc., and chemical physiological investigations on novel types of actions of the endocannabinoids. The ultimate goal of such investigation could be the possible involvement of the endocannabinoids in the chemistry of emotions, a field still in enigma. Um,
That's something he brought up. I've only spoken with Rafi one time through email, but that was something he specifically brought up a couple of times. That he's very interested in understanding how endocannabinoids influence emotion and personality, which I find really really fascinating. That like really hasn't gotten a lot of attention yet.
yeah, I mean, your emotional well-being is directly tied to your endocannabinoid system in a way that you know um, you can imagine that cannabinoids, which can help you lower stress, can actually have beneficial impacts on the body, especially in particular disease states. And I think that like if there's like a kind of like an emotional sort of charge of molecules that like when you feel happy or when you feel sad or when you feel um, disappointed or what, that could certainly be a manifestation of cannabinoid receptor action. And if the mechanism of those interactions are better understood, maybe they could be used to treat anxiety or depression or other behavioral disorders, again, that the brain is trying to overcompensate because it's either been injured or insulted in some way. Um, to me, the endocannabinoid system sort of is the chemical messaging system within the body that regulates all other systems. And so I would think that it should be intimately involved
Yep.
in not only those, but, like he's saying here with inflammation, cannabinoids are very potent anti inflammatory agents and they're also potent antioxidants. And so that points to the use of cannabinoids to control inflammatory disease, traumatic brain injury, um, certain things that are associated with autoimmune disorders.
Yep.
Um, we were just reading a paper how severe stuttering has patients who have taken cannabis oil are helped in stuttering because the stuttering is some type of misfiring in the brain between what the person is trying to say and what they're trying to enunciate. Um, we find things, Jason, all the time, and I'll switch to my next frontier slide, which is that I think, and hopefully, what I was able to share today was sort of like my personal journey as a chemist, wanting to understand the chemistry and the history of the people who came before me so I can do something meaningful at that frontier. Because if you're just repeating stuff that's already been done, you're not really doing anything new, but you're not helping move the entire frontier into that new space. And then 10 years from now, people will be reading my papers and saying, hey, this guy was ahead of his time, or he did stuff that, is now inspiring this new work. Because I think Meshulam recognized, because he did that deep dive and he looked at the the Dunstan and Henry work. He looked at the Kahn work. He looked at the Wood Spivey and Easterfield work. And all of that collectively gave him the knowledge that he needed in order to novelly contribute at that frontier. So I think where the frontier is right now, uh, again, we talked a lot about Delta 8 and those reaction products that are being made, but I think we need to really fully understand and characterize all cannabinoids present in cannabis extracts, whether they're extracted or synthetic preparations. These synthetic preparations, as you know, now create abnormal compounds that the plant doesn't make. Well, what are the impact of those abnormal compounds on human use? We just don't know. And until we have them made and prepared and know that, hey, we need to keep the ISOs below this certain amount, that's what we would use to set a product specification for Delta 8. We would want Delta 8 with, a very minimum profile of these other unknown compounds, or maybe those iso compounds are more active. Maybe they're beneficial. Maybe they add to the overall entourage effect. And so, in a way, we have the opportunity to basically explore both because we have chemistry that can create these isos. Well, why not make them and see if they're, complementary in activity with the cannabinoids?
Yeah, absolutely.
Integration of cannabinoid biosynthesis is a real challenge and something I'm seeing a lot of. Jason, did you have
big money going into biosynthesis? Oh, I was just going to echo what you were saying, and then I was going to say that segues perfectly into the biosynthesis piece, because that's sort of one of my understanding is one of the drives to go to biosynthesis is perfecting, the synthesis itself, trying to eliminate unwanted byproducts and everything, and that sort of thing. So it's interesting to watch, and it's not just the cannabis space too; it's the psychedelic space. There's a lot of money being poured into trying to explore either with yeast or E. coli or whatever, trying to get these microorganisms to synthesize these products theoretically more efficiently than they can do on their own in the lab.
Well, one thing about biosynthesis is that, the plant is biosynthesizing, right? So biosynthesis happens in the plant. I think what we're talking about here for commercial biosynthesis would be genetically engineering a yeast or a bacteria or some other bug that doesn't have the cannabinoid biosynthetic pathway in, putting the pathway in because the genes that code for the enzymes of the pathway are all known. So now that those are known, you could basically create these modified organisms that can now, the way that natural yeast makes beer or makes alcohol,
yep.
You can brew cannabinoids. Well, yeah, I mean, being a cannabis person, I mean, I like my cannabinoids and cannabis myself. I mean, I like the terpenes and the entourage effect that you get from cannabis consumption is probably going to be difficult to reduce to a biosynthetically derived product unless somehow it's like spot on natural with what's going on in the plant. And so now you just have a different genetically modified plant to basically make your cannabis extract. But I would think where cannabinoid biosynthesis would be very important is not sort of at the artisan cannabis product level, but at the pharmaceutical synthesis level, where now products that need to contain cannabinoids that perhaps don't utilize the entourage effect, but still utilize the benefit of having cannabinoids in there for therapeutic reasons, are being incorporated into creams or lotions or other medical preparations, pills or tinctures that basically are targeted towards a particular therapeutic area. You can imagine, again, just like with Sativex, which is, I think, is a spray that's being orally administered to the patients that are predominantly CBD, that similar types of formulations or preparations can be made. And it doesn't necessarily have to have cannabinoids that are derived from cannabis. Those cannabinoids can be derived as an active ingredient from fermentation processes that can be scaled up to very large scale of production. I don't think that will ever consume the cannabis market as we know it, because I think humans still appreciate the flavors and subtleties of cannabis and cannabis products, that I'm not sure if the biosynthesis will ever be something that will be a something that would make cannabis cultivation no longer happen. But I could be wrong on that. I don't know. Who knows? So I think there's a variety of different reasons to take cannabis for therapeutic use. There's the notion that cannabinoids, similar to vitamins, are used to nourish your endocannabinoid system, and that disease state can be described as an endocannabinoid deficiency. So I think daily supplementation in diet and consumables are not going away. I think humans have used this plant for millennia, and until last century, the cannabis deficient century, totally embraced the plant. So maybe again, what's going to happen this century is that the fact that prohibition is going away, and people are becoming more educated on what the properties and potential uses of the plant are, that there will be more exploration of using cannabis as a daily supplement and or food ingredient. I could see CBD being incorporated into a number of different nutritional foods, nutraceutical types, or even fortified into kids' food and geriatric medicine and everything in between. So I think that the final frontier is really understanding that ensemble effect. What Todd pointed out early on, which is that for some reason the resinous exudate extract is much more active than the isolated compounds. I mean, that kind of changes the perspective on single ingredient medicine. And, if you're watching the evening news and you see the commercials that sponsor the evening news, you'll see all of these pharmaceutical commercials that talk mostly about the side effects of these FDA approved drugs. And whatever is wrong with people, the pharmaceutical companies will have you convinced that there's a pill for that. So whatever is wrong with you, you should be taking pills. Well, I think that we need to change our thinking and our understanding of how do we treat disease. And I'm in the firm belief that whenever a medicated herb like cannabis or kratom can be used to treat a disease, that it should not only be lawful, it should be encouraged before taking pharmaceuticals with all their noted side effects.
That's something I've never understood, is why, and I guess maybe it's changing, but it's still really, really prevalent that clinicians will hold back on, kind of toying with the idea of a patient using cannabis until everything else has failed. And it's a weird approach, because cannabis's safety profile and how good it is and everything, Um, and wouldn't it make sense to have that be something you try earlier on to see if that helps, and if not, move towards other things that have narrower therapeutic dosing windows and more side effects, and that sort of thing. And it's been a kind of backwards way that even with clinicians that are trying to get into medical use of cannabis, you're still seeing that pattern play out. That doesn't make a lot of logical sense.
Yeah, I think there's something to that. I think like with clinicians and doctors, I think they're raised in the pharmaceutical model where it's a single active ingredient in a pill, and it's the active ingredient is what's going to knock out the disease. And I think this idea of a polypharmaceutical where you have multiple active ingredients at different levels that are somehow making this synergistic mixture of something that's more active is just something that's difficult for them to get their hands around because of the variability of the plant, number one. I think number two, just as variable as the plant can be in having that effect, the patient's own body and own genetics, how it's predisposed to cannabis. One of the first things I always ask, Jason, when people reach out to me and say, oh, my aunt has got cancer and we want to start giving them, cannabis oil or CBD oil or something. I think one of the things I always ask first is, well, is that individual a cannabis user? Has that
Right.
person ever used cannabis? Are they uncomfortable with the cannabis induced euphoria? I mean, some people are, some
Mm-hmm.
people are not. Some people look for that as being the way of determining that they're actually, that they're getting the medicine. So,
Right. Yeah. Yeah.
um, we have a couple friends one friend who's diabetic who takes kind of like a Rick Simpson preparation, and he does get off on it, but he says, Mark, that's how I know that I'm getting it. Now he claims that, and I have nothing but great wishes for him. Is that Western medicine failed him. Uh, he thinks he actually got diabetes from being treated from other diseases, and that this polypharmacy of FDA drugs that they were pouring down his throat wasn't giving him the level of health that he's now achieved with a full spectrum
Yeah.
oil regimen. Now, I think I always caution people on, I don't think cannabis oil cures everything.
Right.
I
Yeah.
don't. But I think like my wife read something to me this morning over coffee where it was a study that showed double-blind placebo study showed that one brand of CBD oil, all patients in the study lost weight, and they didn't realize they had enrolled in a clinical study looking at weight. And I don't even think they were thinking about weight loss at the time, but weight was just one of the clinical parameters that they followed on this set of patients. And what they found was, I think they were just taking CBD oil and then filling out like questionnaires on general
Mm-hmm.
wellness and are they having any problems with appetite or sleep or bowel movement or anything like that. I mean, any impact on your health negative. But what was noticed across the board is that every patient in the test group, not the placebo group, every patient lost weight. Well, does that say that, a capsule of CBD tincture a day is something that, we should be taking to control weight? I mean, you
Right.
would think that, again, there's like so many different opportunities kind of like at the nutraceutical herbal supplement level that can be explored and exploited at that level, and then ultimately be brought over into the pharmaceutical level where you're looking at, say, 100 milligram pill of nothing but CBD in it, right? Where you're
Right.
just trying to
Yeah.
get CBD into the system. And maybe again, there is some crossover between single ingredient pharmaceutical medicine and the cannabis plant, where, again, this entourage effect is a real thing. There's something really happening that gives greater biological activity to the sum of the parts than the individual ingredients. Nobody understands it. And I think, again, my last bullet point here is if a lab could come up with a way of measuring or better understanding the mechanism of the entourage effect, we might be able to make more effective medicines.
Yeah.
And medicines that are perhaps more active at a much lower, less intoxicating dose. So maybe my friend who takes the RSO, who's looking to feel intoxicated to know he's getting, well, maybe he doesn't need to quite go there all the way if it's in a different form, right?
Absolutely. And I brought this up when I had the chance last year to talk to Dr. David Meary about some of the work that, his labs have done, because they've done some nice characterization work, looking at larger numbers of cannabinoids and terpenes than, a lot of labs do. And, um, in that conversation, it seemed like we're on the path to getting there to some degree. Like, this is such an immensely complex puzzle that when I talked to Vincenzo De Marzo about it, he was like, it'll be at least 2 lifetimes or more before we start to really, piece that together the way that we would like it to be seen. But Dr. Meary, was kind of pointing out that in doing some of just the kind of simple, I say simple, but that's a gross oversimplification of what they're doing. But the sort of like simple sort of assays that they have kind of systematized of breaking up extracts into, they basically break it up into a whole bunch of different fractions, and then they expose those individual fractions and combinations of fractions to different cell cultures and things, and try to come up with an idea of what constituents in the extracts are contributing to what effects, both in isolation and together, in whole combination and in all of these sub combinations. And that type of work, I think, is going to start to be replicated quite a bit by some other labs that understand the value in trying to understand, those dynamics and what it can lead to. And, what he was saying is that he felt like what we'll learn eventually is that, there may be these handfuls of combinations of cannabinoids and terpenes, that drive some of the primary, sort of examples of entourage effects that we see in clinical practice. And that, yeah, when we get to that 1 or 2 lifetimes away from now, we may know that, okay, if you're wanting to treat, let's say, epilepsy really effectively, then you're going to need, you're going to at least need the presence of these 5 cannabinoids in a certain kind of ratio to one another. And you're going to at least need, some of these terpenoids or other chemical constituents, because there's way more than that, in certain relative abundances, to maximize those effects. And it's possible that in our lifetimes, we could get to something like that. Even if we don't understand why they work, we may at least understand some of the patterns that bring about these unique effects that we witness, which is really exciting. There's still a long way to go, but some of those steps are being taken, and it's exciting.
Yeah, well, I kind of liken that to people recommend, like bud tenders recommending strains for anxiety or
Mm-hmm.
or whatever. I mean, I think we were in a dispensary in Colorado a couple of weeks ago, and a patient came in and she was asking about strains that can be used for pain and inflammation. I guess she either had some kind of operation where she was trying to get herself off of painkillers and heard that cannabis can help. And so I heard the bud tender say, Trainwreck and Jack Herrera. And so right away, I start thinking about the terpene profiles, Jason. Yeah, it's like, okay, what's in the terpenes there? It's like, okay, so if those are good for pain, maybe terpinolene, right? Maybe I think this like terpinolene dominant strains could have some kind of impact on pain,
Right.
And then the question is as well, do you want flour? Do you want to concentrate, or do you want a medicated oil or some kind of balm or salve? So, yeah, I mean, I think, yeah, exactly what Vincent said is absolutely true, because you wouldn't think that, again, when you're classically trained as a pharmacologist or as a scientist or as a chemist, you think about a single active ingredient. You don't think about multiple active ingredients. But what I keep hearing too from people about these varieties that have CBD and THC, and you wouldn't think that this would be the case, but because CBD is kind of not really a CB1 agonist,
Right.
but these compounds somehow work in different ways on the enzymes that are
Mm-hmm.
responsible for the physiological effects. And you can't necessarily explain it. You wouldn't say, well, okay, a 1-to-1 CBD THC, you should feel, cannabis induced CB1 agonism euphoria, but you do. And you know, people sometimes make the mistake of making a choice on a cannabis product just solely based on percentage of THC. And I've been schooled on this many times. It has nothing to do with the percentage of THC. Really
Yeah.
doesn't. And it's the percentage of everything else that's in there.
Yep.
And sometimes, again, if you consume something that has a lower amount of THC and it totally just like knocks you out, and then you look at something that's got like 100% pure THC and that hardly moves your
Yep.
CB1 needle at all. It's amazing that you could change this mixture so much and still have that desired biological activity.
Yep.
And getting high is just one manifestation of that, right?
Yep.
So if certainly you're getting that medicine to a tumor, or you're getting that medicine to somebody who's in the middle of an epileptic seizure, it's something completely different. But yeah, I mean, I think, again, just to go back to next frontiers, I think another big challenge for cannabinoids is the fact that they're so darn hydrophobic. They're so sticky to deal with. And
Mm-hmm.
if you've ever had to deal with cannabis flower stickiness on your hands, you know how sticky your hands can get. So that stickiness tends to make dust and dirt stick and germs and other
Yep.
things. So dealing with formulation, cannabis formulations, a lot of people are talking about these nano
Mm-hmm.
formulations and coming up with uh, fast acting formulations that have a particular formulation, because you can't just formulate the cannabinoids in water. You have to formulate them in oil and then make these micro oil droplets and then make sure that it doesn't phase separate. And so there's a lot of chemistry challenges to trying to come up with well, even more trying to come up with something that tastes good. So if you're trying
You're
to
right. Yeah.
come up with a cannabis beverage, you see a lot of these cannabis infused spirits these days. What I find is a lot of them just taste like hell. Taste like the surfactant that was used to solubilize the cannabinoid.
Yeah.
Surfactants are
Soapy.
soap like molecules. So yeah, it's basically like drinking soap. You want to drink soap? So there's challenges there around coming up with a really good water soluble formulation. And the cans that these things come in aren't really designed to hold
Mm-hmm.
cannabis infused beverages. And so a lot of the active ingredient payload is distributed along the surface of the
Liners.
can. Yeah, the
Mm-hmm.
liner of the can, because it doesn't like being in solution. So some challenges there that hopefully chemists will rise to the occasion and come up with some new technology or figure out how to solve these problems.
Yeah, absolutely. It's an exciting time, and part of doing this class, and having you here and laying this out is, curious about cannabis as a whole. My sort of refined mission with all of this as this platform evolves and becomes, this kind of interesting education platform that's becoming, is trying to help people see these frontiers. It's about trying to help get people in that perspective where they can see where things are going or where they might go, or and to be able to ask the best questions possible, armed with that perspective. And so, I know that everything that we dove into here, I imagine everyone going through this lecture, they're probably going to want to rewatch this video a couple of times, because there's a lot to unpack and a lot of studies to, that I recommend really like going and finding. And certainly, if I can get copies of those papers from you, I'll make those available
Yeah,
to the
sure.
students. But there's so much to dive into if you're interested in cannabis chemistry, cannabinoid chemistry. Like this was the high level overview of what's happened and where things are going. But then you start diving into these papers and like honing in on different details that interest you. And there's so many rabbit holes you can jump down where you can kind of get a sense. And I do like the idea of doing something like this for terpenes as well, because it's interconnected and on its own path at the same time. But as you figure out what your interests are in cannabis science and cannabinoid science and everything, and you figure out how you want to leverage an understanding of the science to better whatever it is you do, whether you're working in extraction or formulation, growing, whatever, um, getting, like Mark said, like figuring out the crystal ball, like figuring out the body of work that you need to understand to be able to refer to and figure out those better questions, better perspectives, clearer perspectives, I would say, um, can really help you in all sorts of ways, and have you focus on projects that ultimately in the long run would be more interesting. And that's what always gets exciting to me, is seeing people choosing to do, focus their projects on things that, like you're saying, are more, in that fringe cutting edge, the stuff that the people haven't necessarily taken the time to really tease out for one reason or another. So that's something that always excites me about doing these classes, is hoping that I can, help facilitate that path towards this kind of renewed perspective. As always, talking to you, Mark, this has been awesome.
We could
And
talk another hour. I got more stuff.
I was about to say we could go for another 5 hours.
Well, we'll have another class, Jason. Let's get another class, and we'll get some of these folks to return, and we'll pick this up where we left off. So
yeah, sounds great to me.
I have a lot of enthusiasm for it.
Yeah, absolutely.
Thank you for listening. And Jason, thank you for the opportunity. It was a lot of fun.
Bye-bye.